Hello everyone, this is the GIS Biochemistry Topic 1 review, which is about the digestion and absorption of carbohydrates. Before we get into it, you have to keep in mind that this video is a summary of Topic 1. Use it for revision purposes only. Every single detail is mentioned in the main lecture, so go back whenever you need. Now, let's jump in. First, we need to know about carbohydrates. Carbohydrates are classified based on the number of sugar units and how complex the molecule is into 4 groups. These are monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Monosaccharides are of 1 sugar unit. The most important examples are glucose, which is an aldehyde consisting of 6 carbon units, and fructose, which is a ketone consisting of 6 carbon units. Disaccharides, on the other hand, are molecules of 2 sugar units joined together by a glycidic bond. Examples are sucrose, which is made up of glucose and fructose joined together by an alpha-1 beta-2 bond. Lactose, which is found in milk, is made of glucose and galactose joined together by a beta-1,4 bond. And finally, maltose and isomaltose, which are made of 2 units of glucose joined together by an alpha-1,4 and alpha-1,6 bonds, respectively. Oligosaccharides are carbohydrates of 3 to 10 sugar units. It may be considered as part of polysaccharides that consist of starch, glycogen, and cellulose. For starch, it's a glucose storage form in plants, and it's never found in animal cells. Glycogen is the synonym for starch, but in animal cells. Cellulose is indigestible sugar in humans that is found in the cell wall of plants. Now, let's focus on the most important polysaccharide, which is starch. Starch is said to be a glucans since it's made of glucose. It has 2 types, the long non-branched amylose, which have the alpha-1,4 bond and forms 15 to 20% of starch in granules, and the branched amylopectin, which have the alpha-1,4 and alpha-1,6 bonds, and it forms 80 to 85% of starch in granules. Now, digestion is said to be the breakdown of large molecules into small ones that can be absorbed. The large molecules could be polysaccharides or disaccharides, and they are broken down into monosaccharides. Polysaccharides are further classified as digestible and indigestible sugars. Cellulose, which is indigestible as we mentioned before, is not digested because we lack the beta form of the enzyme amylase, and it bulkens stools and prevents constipation. Other polysaccharides such as starch and glycogen are considered digestible in addition to disaccharides. For fibers, these 2 types have some functions that make them useful. For example, soluble fibers lower cholesterol, improve blood sugar, and you just feel full after eating it. Insoluble fibers, on the other hand, work as a broom, مكنسة, in which they prevent constipation and keep the colon healthy by preventing it from colon cancer. Food will travel the alimentary tract and will face some enzymes that will break it down, preparing it for absorption. These enzymes in sequence are salivary amylase, which breaks starch into dextrin and glucose in the mouth. Next, we have the pancreatic amylase that is produced into the intestine by pancreas. This enzyme has the same action as salivary 1, breaking down of alpha-1,4 bonds but not alpha-1,6 bonds. It does not produce free glucose or other monosaccharides. And lastly, the intestinal glycosidases, which include maltase, isomaltase, lactase, and saccharase, all of which used to break down the corresponding sugar molecule. Some people may face lactose intolerance due to lactase deficiency. It's an autosomal recessive and occurs early during the lactation period. And due to accumulation of lactose and bacterial fermentation, large amount of acids and gases are produced. For the congenital saccharase isomaltase deficiency, it occurs after the child starts to consume fruits and vegetables. Stomach cramps, diarrhea, and other are typical symptoms similar to that of lactase deficiency. After having the monosaccharides ready, many transporting mechanisms are activated. Of these are simple diffusion, which does not need neither carrier nor energy to occur. It moves fructose and pentoses down concentration gradient. Second, facilitated diffusion, which does lead to a carrier with no energy consumption. It moves glucose down concentration gradient by GLUT, which will be discussed later. Third, secondary active transport, which needs a carrier and energy. It moves glucose, galactose, and mannose up concentration gradient. For facilitated diffusion, we use a specialized sodium-independent glucose transporter called GLUT. We have GLUT 1 and 3 with its high affinity, low km, to glucose in the brain and erythrocytes. GLUT2, which is found in the liver, pancreas, and intestines, with its low affinity to glucose, higher km. GLUT4, which is unique in its dependency on insulin. It's found in skeletal muscles and adipose tissue. GLUT5 is specialized for fructose and found in the intestinal cells. About active transport, it is achieved by a sodium-dependent glucose transporter with NA-K pump consuming energy. It is secondary active transport. SGLT1 is found in the enterocytes and proximal straight tubule, while SGLT2 is found in proximal convoluted tubule and contributes to renal glucose reabsorption. This method is used to transport glucose, mannose, and galactose. After absorption has occurred, galactose and fructose mostly converted to glucose. Glucose is catabolized for energy, which is the preferred energy for most of the body tissue, and it's catabolized by glycolysis, Krebs cycle, and electron transport chain or oxidative phosphorylation. Glucose is stored as glycogen when it's in excess, and glycogen is stored in the liver and muscle mostly. We use glucose also, or other monosaccharides, to synthesize other carbohydrates, like fructose for semen, galactose for lactose synthesis, pentoses for nucleotides. We use glucose also and other molecules to produce non-essential amino acids, and these non-essential amino acids are produced in Krebs cycle. We use it also in lipogenesis, which is in the synthesis of glycerol 3-phosphate. Also, we use glucose to synthesize acetyl-CoA, which is converted into fatty acid, cholesterol, and ketone bodies. And that's why glucose is not beneficial in the hyperlipidemic cases.